Hi again, everybody. Um, Today we are starting a little bit earlier with our journal club because, as you can see, we're going to present one of the work from one of our Chinese users, Lei Guo, who's doing his PhD in Shanghai right now. And my name is Elena. I work as a scientific consultant here at NIRx. And before we start, I would like to give you a little bit of background on us and how to reach us. So as you can see here, this webinar will be recorded and will be found on YouTube on our YouTube channel, but you can reach out to us through consulting@nirx.net and through more or less all the social media. If you want to be updated on our latest news and all the webinars we're organizing, journal clubs and so on, please subscribe to our newsletter. And so without more introduction, I would like to present you to Lei Guo, who's today our speaker. He's doing his PhD, as said, as a member of the addiction study group at Jiaotong University, the School of Medicine in Shanghai, China. He has a major in psychiatry, and his main research is on pathologic psychological mechanism and physical intervention of substance dependence. This is his link to his publications. So if you're curious, you want to check it out. And I would like to give you a little bit of background on the Zoom call here. So you will all be muted, but please feel free to unmute or to write your question in the chat at any moment. The content will be available on our website. And if you have any questions, you can write us to consulting@nirx.net or support@nirx.net. And without further ado, I will leave the virtual mic to Lei.
Okay, and I will share my screen. Yeah, can you see that?
Yes, we can.
Okay. Many thanks to the introduction by Elena. And I'm mainly working on the mechanism of addictive disorders. And I give them with physical interventions like TMS or TACS. And this study is conducted about 2 years ago and it was published earlier this year. It's a fNIRS hyperscanning study on the alcohol use disorders. So let's get started. Let's talk about a little about the background. We all know that alcohol use disorder is a very severe economical and health burden on the world. In China, according to a survey several years ago, the prevalence of AUD in males have reached about 10.1%, is pretty high. And there is also a thing called white wine culture in China. In many regions, people like to use wines to express their emotion or use to make friends. It's just like a social lubricant, especially in the northern part of China. So in these regions, the prevalence of AUD is particularly high. And we all know that there are many negative consequences of AUD. They will affect multiple organs, and they may also cause some mental problems, like changed emotional behavior and cognitive dysfunction, and more seriously, they may cause Cossack syndrome. People get have some problem with disorientation. They have a loss of memory or making up stories because they can't remember what they have done before, and they start making up stories. These are very serious consequences. But what really caught our interest is that some studies have found that the echo may affect some key process of social interaction. At a meta-analysis in 2017, have found that there are some facial emotion recognition deficits in the AUD patients. And also they may have some deficits in the theory of mind. A theory of mind is the ability to infer the intention of other people. So it's also a very important ability in the social interaction. So we want to get deeper of this topic. But the problem is, how can we measure the social function of a subject? In the traditional way, we can use the person-computer interaction. We will ask a subject to play a task related to the social conditions. In these conditions, people actually are playing as a third person, observing the social condition we provide to him. So this kind of task is has a lower ecological validity. However, some researchers design the tasks that they will ask 2 or more subjects to complete the task together. And in this process, the subjects are really interacting with each other. And if we measure the brain activity simultaneously, this kind of design is called hyperscanning. So we are actually measuring the multiple interacting brains. So we can interpret how social interaction is happening. And some tasks are very classic, like eye-gazing task, like imitation task, or cooperation competition task, which we used in our study. And the first hyperscanning study was conducted in 2002. The researchers put 2 subjects in the 2 fMRI scanners, and they are labeled as sender or receiver. And the sender will see a colored square, and he can choose to tell the real color he see to the receiver or not. And the receiver have to guess whether the sender is deceiving him or not. So in this process, the 2 subjects are really interacting with each other. And interestingly, the researchers found a significant coherence in the supplemental motor area during the task. So this means that the brain activity of these 2 interacting subjects become similar when they are interacting. So this is very interesting. And when we are designing a hyperscanning study, there is a very important hypothesis, is that the interbrain synchronization is a mechanism of social interaction. So many studies have found that the interacting people, their brain activities become very similar. So the ABS reflects a dynamic interaction between the neural states of socially interacting individuals. This phenomena have been seen in many studies, like mother-child interaction or joint social decision-making. They have to make decisions together, and even some clinical psychotherapy between the psychotherapist and the clients. So this is very interesting. And we also want to do some studies like this. And we have to think which kind of tool is better for hyperscanning. We have many choices in the previous studies. They use fMRI, they use EEG or MEG, and fNIRS. But after the investigation, we found that fNIRS is particularly suitable for hyperscanning because it has a much better temporal resolution than fMRI, and it also have good spatial resolution. And also it's very robust. to motor artifacts. If you use EEG to record any tiny movements will cause a large fluctuation of the signal, but fNIRs is more robust to the movement. So we are prefer to use this. And also fNIRs is very easy to set up. You don't need to use gels or any other things. You just put the cap on the head. So it's very easy to set up. And for this reason, it's very compatible with multiple populations, including patients or children, etc. So this is our study. And if you want to know more details about the study, you can check this paper. It's already published. And I will have a brief introduction of our study. We recruited about 28 subjects with AOD and 36 healthy controls. And we paired them into 14 AOD dyes and 18 healthy control dyes. We have we use some methods to make their age and age differences comparable, so that there will not introduce any other influencing factors. And here is the inclusion and exclusion criteria. And the criteria is the patient with AOD must meet the DSM-5 diagnosis system and around 18 to 60 years old, and have no other psychiatric diseases or other physical or neurological diseases, and they can fully understand the items of the questionnaire and the request of the tasks. And if people have drank so many wines, and their cognitive function can be very bad, so we choose the ones that can fully understand our tasks. And we also have some clinical measures. We measure their drinking status with alcohol use disorders identification test. Is widely used questionnaire to measure their levels of drinking. And also we use the PH9 and the JD7 to measure their depression and anxiety levels. And we also use the Barrett impulsivity scales to measure their impulsivity, because impulsivity is also very common in the alcohol users. And here is a behavioral task used in our study. This task is called the cooperation competition task. It's first developed by Professor Xu Cui, who is giving lectures in Stanford University. And yeah, he's a Chinese. And this is also the first hyperscanning study with fNIRS. This study is quite simple. It requires two subjects set separately and facing two separate screens, and they could not see each other's hands. And this task have two conditions. In the cooperation condition, the subjects was instructed to press the keyboard as simultaneously as possible when the gray circle was filled with the solid green circle. So this gray circle appears, the subjects are reminded to focus, and when the green circle appears, they have to press their keyboard simultaneously. And in the competition task, the subjects have to respond as fast as possible when the green circle showed up. They don't care about the other one's response. They just press the keyboard as fast as they can. So in this process, there's no interaction between the two subjects. And also we set a threshold. It's a 1/8 of the sum of two the reaction times of the subjects, because two subjects can never press the keyboard exactly simultaneously. So we set a threshold. If their reaction time difference is lower than the threshold, we will consider them pressing the keyboard simultaneously. So this is a task. And also when they are playing the task, we use the fNIRs hyperscanning. We use the NIRx Pro 2 fNIRs recording system. This system have independent hyperscanning module, so we can perform the task very easily. And we use the two wavelengths of the fNIRs with the sampling rate around 12 Hertz. About the montage, we put 8 sources and 7 detectors on the prefrontal of each of the subject. We know that fNIRs measures the region between the sources and the detectors. So this kind of setting will consist of 20 channels. And the opto's position is accordance with the 10-20 standard EEG montage. So you can easily convert the opto's position to the brain coordinate and to do further analysis. And here is the data process. It can split to 2 parts. The fNIRs pre-processing include convert the raw intensity to optical density, and then we will correct the motion artifact, and then convert the optical density to the concentration of HBO and HBr in the brain. And then we can use the processed data to calculate inter-brain synchronization. We used a generalized wavelet coherence method, and to increase the robustness of our results, we use a bootstrap procedure to calculate a cutoff value, and any IBS value higher than the cutoff value is considered significant. And then we calculate the significant ratio. This ratio was used as a dependent variable in the further analysis. And also we used a validation procedure because there may some random synchronization between 2 brains. So we permute the subjects in a pair and compare the real IBS between the real pairs and the permuted pairs to select the real the IBS that is significantly higher than the chance level IBS. And then we used a GLM model to calculate the significance of the effects we are interested. It's quite easy. We use the IBS ratio as a dependent variable, and we included group and task main effect and their interaction item in this model. And we do this model across all the channels. So there are 20 models, yeah. So we can see that the audit score of the AOD pair is higher than the healthy control. That means they have a much higher level of drinking than healthy controls. But their age and age difference in the pair is not significant. So, and this is the main result of our study. So you can see this picture is a demo of the wavelet coherence of the collaboration task. Maybe you have seen it in many studies. The x-axis is the time, is the time in seconds, and the y-axis is a period, is a reciprocal of the frequency, and the horizontal line indicates our interested frequency band in this task, and the vertical lines indicate the task blocks. You can see there is a little rest between the blocks. So we can notice that the IBS in our interested bands has an increase. So this means in this task, their brain is synchronizing with each other's. We can also see an increase of IBS around 1 Hz frequency band, and this is caused by the heartbeats and breath, these physical brain blood fluctuations. So this is not what we're interested in, and we just interested in the IBS in this frequency and time bands. So we can see there is a significant main effect of group and task effect in channel 20, and this is located in the ventrolateral prefrontal cortex. And then we conducted a post hoc analysis, and we found that in both cooperation and competition condition, the patients with AUD, they all showed a decreased IBS compared to healthy controls. And this decreased IBS during cooperation task is correlated with the increased non-planning impulsivity measured by the Barrett impulsivity scale. So this is a result of our study. And we infer that the disrupted function of VLPFC is very common in the patients with AOD because it's consistent with the previous studies. And we infer that the structural and functional profiles in AOD may lead to disrupted cognitive control and social information processing functions, and then affect their synchronization abilities of prefrontal neuron activities. And this will cause a decreased social abilities. And from the correlation analysis, we infer that subjects with high non-planning impulsivity may focus less on the feedback of the task and have difficulty in adjusting themselves for the future cooperation trials. So this is our discussion, and this study has some limitations, including this cross-sectional design. It cannot conclude any causal inference. And this study has a small sample size. It's also affected by the COVID-19. And we also did not collect the AUD and healthy control pair. The pair is just AUD to AUD and healthy control to healthy control. The AUD healthy control pair is more close to the real life of the patients. So it's also a limitation. And in the end, sorry for the timeout, but in the end, I want to discuss a little about the IBS. Although we hope that IBS is the mechanism of the social interaction, but some researchers think that IBS is just a byproduct of the social interaction. It can come from what we observed. The synchronization can come from multiple reasons. Like the brain activity can be similar because of they are receiving the same external stimuli like sounds or images, or maybe they're just because they are both talking, or maybe they are just have paying attention to something simultaneously, so their brain activities become similar. So this is very important. This tells us when we want to design a hyperscanning study, we have to carefully design our task to rule out all these potential influencing factors. And the best way to prove whether IBS help social interaction is to design some causal studies. We can use the neuromodulation techniques like TACS or TMS to intervene the interacting brains. Actually, there are already some preliminary studies on this topic. So if you are interested in this topic, you can check the papers below. There are many wonderful discussions on this topic. And okay, in the end, I want to express my acknowledgement to my team in Shanghai Mental Health Center. And also, we want to thank the NIRx company to provide such a wonderful device so we can conduct our study without any technical barriers. Actually, it's quite difficult if you want to set up such a system by yourself. So thank you. So this is the end of my talk. Sorry for the typo. Okay. And I'm glad to answer any questions. Thank you.
Thank you very much. So is there any questions from the audience? Otherwise, we received some questions during the registration and I can read them. All right. So someone asked, what software for data analysis would you recommend to use with the type of research presented?
Yeah. What the software I used in this study is the Homer 3 software. It's a open source software that we can download from the website based on the MATLAB. It's very easy to use with a nice UI. So you can just click your mouse to finish your process analysis. But the IBS calculation, you need to write your own script. And if you want to perform more complex analysis, you have to write your own scripts to do it. Yes.
Thank you. Then second question. In your paper, you present participants between 18 and 60-year-olds. Did you find any age-related differences?
Yeah, we controlled this to avoid additional influencing factors. So there's no, sorry, this table. There's no age difference or the age difference inside the pairs between the groups. Yeah.
Perfect. And last question. Do you expect any restructuring in connectivity between different brain areas? For example, somatosensory and prefrontal cortex. Are you planning to expand your head coverage?
Yeah. Brain connectivity analysis is also popular in hyperscanning studies, but we didn't do it because the brain region we covered is small, and they are also they are all prefrontal cortex, so is not so insightful to do such analysis. But if we use whole brain coverage, we can see the interaction between the brain regions when we are interacting with other people. And there are also many techniques or methods to analysis the connection between regions, like grandeur causivity. It can help you to infer some causal inference between subjects or brain regions. And about the coverage of the brain regions, that's because the fNIRS optos are really expensive. If we want to cover the whole brain, the cost is very high. So we have to consider this when we are designing the study. Yeah, thank you.
Thank you. So those were all the questions that we received during the registration, and I don't see any questions in the chat. So I want to thank you again for this very nice presentation, and see you next time.
Yeah, thank you.